Hello everyone, and welcome to our new video. Today we'll be talking about micropipettes, how to properly read and use them. So here you can see a P1000 pipette. So the number after the P tells you its maximum volume. The window here shows you the volume that's currently set to pipette. Here this is 1,000 microliters, so first the top number is the thousands digit. Second one is the hundreds, third one's the tens. This differs from pipette to pipette. And you can actually change this number. So here, it's been changed to 750 microliters. Now you don't, as I said before, you don't want to go above the number after the P, that's a max. So this knob here, when you turn it as demonstrated, it will actually change the volume that the pipette is set to hold. So here you could go up to 1,000 microliters, but also go below that. This is a P10 pipette, so it holds maximum 10 microliters, which is the setting here. The 0 at the end, that's in red, is the first decimal place. The 1 is the tens, and then the middle one is the ones digit. And again, this can be changed, so here it's now set to 7.2 microliters. As you might notice, this is a little more precise than the P1000, but it also holds much less. Now, when you're holding a pipette, you want to keep your 4 fingers in a grip underneath this little overhang, and then your thumb on this button, which is used to draw in and expel solution. There's another button that is responsible for releasing the tip after you're done pipetting. And we'll talk about the tips and how to actually use the pipette now with the P1000. So the tips are to actually contain the solution. You don't pipette, or the pipette itself doesn't take in the solution. That would damage it. These tips are sterile, so you stab them while they're in the box with your pipette, and you don't touch them with your bare hands. And you want to keep the box closed as much as possible. So, when you're pipetting, you want to first press the button down before inserting it into the solution. It has 2 stops. The first stop is for drawing it in. The second stop is for actually expelling. So you push it down once, put in the solution, slowly draw it in, and then when you want to expel the solution, you'll go to whatever receptacle you're using, push it down once to expel the liquid, and then a second time to expel excess air. And this will ensure that you push out the total volume that you drew in. Now we'll show you with the P10. So again, you put the tip on. You'll notice there's a different tip. Different pipettes use different tip sizes. And here we'll actually show you the button so you can see the stops. So what you want to do is push it down once to the first stop, slowly draw it in, go to your receptacle. You can see there's the solution in the pipette tip, and then in your receptacle you will push it down once for the liquid and then the second time for the air. Now when you're expelling, it's actually good to have the tip against the side of your beaker or whatever it is, as this helps draw out the liquid. Another thing you can do with pipettes is actually mix your solution. If something like vortexing might damage whatever you have in your sample due to shearing forces or whatnot, you can use a pipette to just draw it in and expel, and this will mix your solution. Just like when you're pipetting normally, you 1 stop to draw it in, 2 stops to expel it, and then you release the tip. So there are many different kinds of micropipettes. Here they will be in order of smallest volume to largest from left to right. So on the left we have a P2, which would be 2 microliters. And then we have a P10, which is 10 microliters. We'll go over the different tip types in a second. Then you have a P20, a P100, P200, and a P1000. So due to the different sizes of the pipettes, they will have different tips. Some of them share tips if they're close enough in size. And there are also some general purpose tips, which we'll show in a second. So these tips are for the P2s and the P10s. The next set of tips here are for the 20s and 200s. These are actually general purpose, so they can be used for a variety of pipettes. This is for the P100s. And then finally, the large ones are for the P1000. Well, we hope you enjoyed our video on the basics of pipette use. Please leave a like, comment, and subscribe, and we'll see you next time.